Hello. Thanks for all of you for joining today, and thanks for giving me the opportunity to present my research. So let's see. So you should be seeing the presentation right now, right? Okay, awesome. So today I will be talking about the role of estradiol in male depression. We mainly use.
We're seeing the slide view rather than the presenter view rather than the.
Okay.
Excellent.
Now?
Excellent, great.
Awesome. Yes. So first of all, why is this important? So we know in humans that decreased testosterone production, which is called hypogonadism, results in increased incidence of depressive disorders. And if you see here in this cumulative report, what they show is that men that they have normal hormone levels, testosterone levels, they have compared to the hypogonadal men, they show higher incidence of depressive illness. Hypogonadism is affecting around 23 to 39% of the population. Usually affects middle-aged or older men due to aging, but also you can have younger populations because of testicular cancer or obesity or diabetes. So it's not only concentrated on older generations. So a couple of years back, they published this meta-analysis study. So far in the field, there were some reports that they were reporting positive effects of testosterone. Other ones, they didn't find any effects. So seeing all these studies together, it was very helpful to actually draw some conclusions. So here they summarize all the studies, the effectiveness of testosterone treatment in all the studies that they mainly provided in aging populations. So what you see for the effectiveness here of the testosterone is that some of the studies, as I mentioned, they have very positive results, like here, these two, the first two studies. Some of them, they are kind of in the middle. So it was kind of a mixed message. And then when they also look at the efficacy estimates of the testosterone treatment, again, it was something like not conclusive. So you mainly have like these efficacy studies. They show that yes, testosterone might actually be effective for treatment of depression, but then also there are some studies that they didn't find any effects. So what we know so far is that probably the studies that they gave higher doses, really high doses of testosterone, they did show testosterone effectiveness in decreasing depressive symptoms, but then studies that they've used lower doses for shorter periods, they didn't find any effects. And here they plotted like a correlation study between effectiveness and efficacy. And on the upper right hand here, you see the studies that they had good effects both on effectiveness and efficacy. However, as you see also, you have studies like kind of in the middle. Some patients, they showed improvement. Some of them, though, like of the studies, they didn't find any effectiveness of testosterone. So the conclusion from this is that testosterone replacement therapy may be effective for the treatment of depressive symptoms or depression in men. But also we have to consider that chronic testosterone treatment, especially at high doses, these studies suggest that might be the best choice. We have potential serious side effects, including cardiac and hepatic dysfunction, as well as many others. So what we know so far for the metabolism of testosterone is that it's producing the gonads, and then it metabolizes, it exerts its effects three different pathways. The first one is through its metabolism through 5-alpha reductase to hydrotestosterone and then binding to the androgen receptor. Testosterone itself can bind directly to the androgen receptor. And then the third pathway that I will be focusing mainly today is through the actions of aromatase, testosterone is converted to estradiol, and then we have the binding of estradiol to the estrogen receptors, ER alpha, ER beta, and the G-coupled and G-coupled estrogen receptor, which was more recently found in the last couple of years. So in this study, we've used a subthreshold social defeat stress, which is a very mild stressor. And we've used this model as a way to reveal the phenotype in these hypogonadal subjects. So this protocol, what it includes a physical attack phase for 2 minutes from a bigger aggressive mouse, the white mouse, and the experimental mouse is the black mice in this occasion. And then there's psychological sensory stress, which we moved the mouse, the experimental mouse, to the next department. However, we have a perforated divider between in the cage, so the mouse can still sniff and see the aggressive mouse, but it cannot be physically attacked. So I included a video so you can actually see what is going on. So you can see that the aggressive white mouse, it attacks the smaller experimental mouse. And in humans, we can a lot of people, they do associations between this model and maybe bullying or other kind of stress in society. So this is repeated 3 times, and as I will show you later, control mice, they don't show any deficits after this mild stressor. So we test these using 2 different protocols in the current project. So the first one is the social interaction, which mimics the social avoidance that we expect to see in depressed humans. So we have 2 chambers. The one includes a stranger mouse. And you see that the resilient mice will go and sniff the mouse because it's rewarding for mice to interact with other mice. However, in the case of the susceptible mice, the phenotype that we see is that these mice, they are not sure, so they try to interact initially, like here, but then they decide that, you know what, I prefer to spend my time on the empty chamber rather than in the social chamber. And the next test we've used is the female urine sniffing test. So this is an anhedonia-based test based on sexual behavior. So this is an image from the actual test. And the mouse is presented with a Q-tip, a swab from male urine and a swab from female urine. And what they've shown is that actually exposure to female estrus, female urine increases dopamine levels in the nucleus accumbens, which suggests that actually, yes, exposure to estrus female urine in mice is rewarding. So the first question we wanted to ask is, as I mentioned, we have 3 different estrogen receptors. In this study, we concentrated mainly on the classical receptors, estrogen receptor alpha and estrogen receptor beta. And so these mice, we've used conventional knockout mice. So these mice, they lack expression of estrogen receptor alpha. And they went through stress. And what we show here is that after stress, estrogen receptor alpha knockout mice, they don't show any social preference compared to the wild type mice that went through stress, or the non-stress mice and the knockout mice. So we know that we were able, like estrogen receptor alpha is involved in the development of social avoidance. However, we didn't see any anhedonia development in these mice. When we tested the estrogen receptor beta knockout mice, what we've seen is that these mice, they show social avoidance as well as anhedonia following stress, suggesting actually that estrogen receptor beta might have a bigger role in the development of depressive related symptoms. So from now on, we choose to go deep and investigate the exact role of estrogen receptor beta in male stress susceptibility. So the first question after this is, so if we orchidectomize mice, so if we remove the gonads, so we don't have any testosterone production, and consequently we won't have any estrogen production in our male mice. And these mice, they undergo the subthreshold social defeat protocol, and they receive estradiol either after the stress or before the stress, and then tested in these behaviors. Will we see a reversal of this stress susceptible phenotype? And what we've seen here is that we've seen a reversal in the treatment of estradiol in the mice that they received estradiol only prior to the stress for the social interaction. However, we've seen a reversal in the anhedonia phenotype in mice that receive estradiol both after and before the stress. So this suggests that actually estradiol might have some, definitely can be used as a prophylactic therapy for patients that we know that they suffer from hypogonadism. However, with some symptoms, it can be also used in treatment as a treatment for patients that they already develop depression. Okay, so the next question that we wanted to address is, so what is the circuit, brain circuit that's involved in this effect, estradiol effects and estrogen receptor beta effects? We know that from the literature that in human and both preclinical literature that there is a huge role of the nucleus accumbens in the development of these depressive phenotypes. So we've used this genetic approach. We've used the estrogen receptor beta Cre mice. So these mice express the Cre in all the neurons in the brain that also express estrogen receptor beta. And we've used a retrograde adeno-associated virus. So what it does when we inject this virus in the nucleus accumbens, it will retrograde and basically travel back in all the regions that they have estrogen receptor beta projections to the nucleus accumbens. So what we found is that when we did this experiment, we found a huge expression, so a very strong projection, estrogen receptor beta projection from the basolateral amygdala to the nucleus accumbens. And you see here, we have very strong expression in all the different bregmas of basolateral amygdala. So we decided to go ahead and investigate whether this circuit is actually involved in the development of depressive related symptoms in orchiectomized mice. So first, we wanted to address whether the circuit is activated following stress in mice that they receive either oil, so the control treatment, or they receive estradiol. What we did is, again, these mice that received the retrograde adeno-associated virus, the same virus that I've used in the previous experiment, and we allowed 3 weeks for expression. Then they were orchidectomized to remove the endogenous estradiol levels, and then we allowed for the hormones to clear out for 10 days. And these mice received via color estradiol treatment, and then 45 minutes afterwards, they went through the acute stress. And then we collect these tissue samples 2 hours after the estradiol treatment, and we've used this tissue for CFOS, which is a proxy for neuronal activation. And what we've seen is that mice that they receive estradiol, we've seen higher activation of the BLA to nucleus accumbens estrogen receptor beta neurons in mice that they receive estradiol, suggesting that this circuit has actually a role in the hypogonadism and stress-induced susceptibility in male mice. To further investigate this, we decided to use optogenetics. I'm going to describe these techniques for folks that they have more clinical background. We've used these specific proteins, which is packaged in an adeno-associated virus we call opsins, and they are proteins that can be activated through light. And what we attach a promoter region. So doing this, we can actually specifically target specific neurons. So we can express these proteins in specific neurons of interest. And also these proteins, they are attached with a reporter M-cherry or YFP, as I will show you later, so we can see the expression in the brain. So when we do this and we shed blue light, we can activate these neurons or deactivate depending on what specific opsin we have used. So what you see, this is similar to the electrical stimulation, traditional electrical stimulation, but with the electrical stimulation, when you apply the stimulation, you basically stimulate all the neurons in the region of interest. When we have light stimulation without any expression of these proteins, then we don't get any activation. However, when we have expression of these proteins in our neurons of interest and we shed light, then we see activation only to the neurons that express generodopsin. So using this technique, we wanted to address whether activation of the ER beta expressing BLA to nucleus accumbens projections are rewarding. So these mice received generodopsin in the basolateral amygdala. So this is a Cre-sensitive generodopsin, and we're using ER beta Cre mice. So that was to make sure that we will get expression specifically only to ER beta expressing neurons. And then we implanted the optical fiber in the terminals of the nucleus accumbens. So using this approach, we have a very specific manipulation of only the ER beta expressing neurons from the BLA to nucleus accumbens. So what we did is, following expression and recovery of the mice from the surgeries, we use a real-time place preference protocol. So the mice we've used this two-chamber box, and we assign a box aside of the one box that the mice, every time that they will enter, they will receive a light stimulation. So if this light stimulation is rewarding the mice, we choose to spend more time in the light activated compartment compared to the non-light compartment. And in these images, I demonstrate the expression that we get from injections from generodopsin. As you see, we have a clear punctated the labeling on the soma of these ER beta expressing neurons, and then we have terminal expression in the nucleus accumbens where the optic fiber was implanted. So what we've shown is that mice that received light stimulation compared to control mice that they didn't receive generodopsin, but they still receive light stimulation. We've seen that generodopsin injected mice showed preference to the light pair compartment, suggesting actually that yes, activation of this projection is rewarding. Then following this, okay, we know that it's rewarding, but if we activate this specific circuit prior to the stress in these hypogonadal mice, will we see any reversal of the social avoidance and anhedonia phenotype that we've previously demonstrated in orchiectomized mice or ER beta knockout mice? So to do this, again, these mice receive generodopsin or YFP injections, and we allow 3 weeks for expression. And then we're going to tectomize these mice and allow 10 days for clearing of the hormones from the system. Then prior to stress, they received a 45-minute for heart stimulation, a light stimulation. And then on the next day, we tested them on the social interaction, and on the following day on the female urine sniffing test. So what we've seen is that these generodopsin activated mice, so it's the last group here that they receive light stimulation prior to stress. We've seen this increase in interaction with the stranger mouse compared to the YFP injected mice that underwent through stress, suggesting actually that, yes, actually light stimulation activation of this circuit might be involved in social interaction deficits following stress. However, we didn't see a reversal in the anhedonia phenotype using this light stimulation, suggesting that we might have a different circuit involved in the anhedonia phenotype compared to the social interaction deficits. So following these results, we reached to the hypothesis that then absence of estradiol might induce decrease in the activity of the ER beta neurons from the BLA to nucleus accumbens, which consequently after stress can result in the behavioral phenotype that we've seen in social avoidance. So to investigate this, to measure network activity, we've used the hyperphotometry protocol. So in the hyperphotometry, how it works is basically fluorescence acts as a representation of neuronal activity. So in GCAM that we've used in this occasion is a genetically encoded fluorescent indicator. And when we shed light, we get excitation of this GCAM, and then in turn emits its own light, which we record as a proxy for neuronal activity. And we do this through different lenses, and this is a basic setup for the system that we use. So what we get usually from using this technique is traces looking something similar like this. So the blue line is the aciosbestic signal. So it's more like a background, and to make sure that our signal, our recording is stable. And then we get the excitation signal, the emission signal, which is represented in the blue line. And here, for example, we have this bigger peak, which will represent an actual signal detected activation of this network. And when we applied Z-score and we control for the aciosbestic signal also, and the smoothing of the curves, if you see here, this is the same trace as the one that I've shown you before. But you could see that this is what we are looking for in terms of something that represents neuronal activity from our measurements. So using this approach, this technique, again, these mice, they receive the calcium indicator, clay-sensitive calcium indicator in the BLA, and then we use the optic fibers implanted in the nucleus accumbens as a means to activate, to excite these projections, and to also record the signal emitted by GCAM expression in the terminals. So what we've seen in the sham mice, so these are mice that underwent through the same the surgery, so they underwent through anesthesia, but they are gonatically intact. What we've seen is that during social interaction, we have this increase in neuronal activity of the ER beta neurons from the BLA to nucleus accumbens when they interact with a stranger. However, we have a decrease in the neuronal activity when the mouse is interacting with the empty cage. In the case of the orchiectomized mice, what we've seen is that basically these mice, this network is not activated following interaction with the stranger mouse or with the empty cage. So we don't see any difference between the interactions. And then when we compare the areas under the curve between the gonadally intact mice and the orchidectomized mice, we could see a clear difference between the two different phenotypes. Also, this is the scores from the social interaction from the same mice that we recorded. And what we've shown is that, yes, we've seen this preference in the gonadally intact mice. However, we didn't see the same preference in the behavior in the orchiectomized mice. And when we do correlation between the signal, so the activity measures and the actual behavior, what we've seen is that there is a close to significant correlation in the sham mice and the interaction signal. So the higher the interaction time, the higher the signal from the ER beta neurons is. And we've seen a significant correlation between the interactions, the neuronal activity signal, and the interaction with the stranger mouse, suggesting actually that, yes, we don't see two different completely phenotypes. So whatever is happening to the brain in this network is actually reflected in the behavioral phenotype too. So kind of a mid-presentation conclusion. So what I've shown you so far suggests that estradiol through estrogen receptor beta controls neuronal activity of the basolateral amygdala to nucleus accumbens circuit, and that underlies the development of stress-induced social avoidance in these mice. So the next question is, so we dissected the circuit of this phenotype. However, in humans, we wanted also to know what can be used in terms of treatment. And in this occasion, for this reason also, because it's highly unlikely that in humans they will receive one acute treatment of estradiol or any other treatment, we switch our administration protocols to chronic treatments. So the first experiment we did was with testosterone. So we wanted to demonstrate to see whether testosterone supplementation replacement therapy will reverse these stress-induced phenotypes that we've seen. So what we done, the mice were orchidectomized or received sham surgeries, and then they received subcutaneous implants, either empty implants or implants filled with testosterone. And then following 10 days, these mice, they underwent the acute stress protocol and then tested for the social interaction and the female urine sniffing test deficits. And this is the basic also overview of what I've described before. So we have the physical stress for 2 minutes and the sensory stress for 15 minutes repeated in 3 times. And then the mice on the following day, 24 hours after stress, they tested on the social interaction, and the following 24 hours, they tested for development of anhedonia. Okay, so what we've seen here is we have a lot of control groups, but I will draw your attention to these 2 groups. So mice that were orchidectomized and they didn't receive any testosterone, we've seen this lack of social preference. However, when testosterone was supplemented back into the system, we've seen a complete reversal of this no preference phenotype. And we also have seen something similar with the female urine sniffing test with testosterone replacement therapy, reversing the phenotype induced by the stress and orchiectomy. So we can conclude that testosterone was effective in reversing these maladaptive behaviors. So to go one step further, as I mentioned in my previous slide, testosterone exerts its action through the binding on the androgen receptor. So our hypothesis here is if that these effects were really through testosterone and not through estradiol, blocking the androgen receptor in gonadally intact mice, then we'll be able to induce susceptibility, stress susceptibility following stress. So we followed a similar design as a study with testosterone. So these mice, they received chronic treatment with an androgen receptor inhibitor. And then what we've seen is that the supplementation, so blocking of androgen receptor didn't induce any maladaptive behavior. So we've seen that all the different groups that we had here, they showed preference to the social chamber as well as preference to the female urine, suggesting that actually testosterone might not be the main mechanism for the reversal that we've seen previously. So using the same approach, this time we've used an aromatase inhibitor. So aromatase is the enzyme that converts testosterone to estradiol. So using that, we will still have in these mice production of testosterone, and they will have normal regulating levels of testosterone. However, we won't have any production of estradiol. And when we did this, what we've seen is that in this last group here and here, that we were able to induce a susceptible phenotype in these in the mice that they didn't have any estradiol produced, suggesting that the effects that we've seen is not through testosterone per se, but is more through its conversion to estradiol. And then asking the obvious question, if we actually supplement with estradiol, will we've seen this reversal of the maladaptive phenotype induced by the combination of hypogonadism and mild stress in these mice? And we did a dose response here because we didn't know the exact dosage that we should have been used in male mice. And what we've seen in the social interaction protocol is that we've seen the same susceptible phenotype in orchiectomized mice that they didn't receive any estradiol. However, mice that they received the high dose of estradiol, we've seen this full reversal of social preference. I have to note here that we have some effects also of the 3 millimeters if you compare directly the interaction ranges, but it's not quite significant. However, like in the female urine sniffing test, again, we've seen something similar with a complete reversal of the maladaptive phenotype at the medium dose and at the high dose, suggesting, yes, that estradiol is actually effective in reversing male stress susceptibility. However, estradiol as treatment, it has some drawbacks in terms of human consumption that we know that estradiol can have a lot of undesirable peripheral effects in the male population, including gynecomastia, erectile dysfunction, and others. So it's not an ideal and a viable treatment option for the male population. So what we use here, our collaborator developed this new compound, which we call DHET for a very long name, and it's converted through an NDPH conversion. It's converted to estradiol, but this conversion only occurs in the brain and not in the periphery. So using these two compounds, we can have the brain selective effects of estradiol without the unwanted peripheral effects. And as a confirmation, we performed a PK study, and when we injected D3 estradiol in male mice and then measure estradiol produced, when we injected the D3 DHET in these mice and then measured D3 estradiol, what we've seen is that we've seen this huge increase in estradiol production from DHET in the brain. However, we haven't seen any production of estradiol in the periphery, actually confirming that yes, DHED can use as a tool compound for brain selective estradiol delivery. And then when we've used this compound for in our behavioral paradigms, what we've seen is that similar to the estradiol treatment, we've seen complete restoration of the social preference behavior as well as restoration of the anhedonia phenotype, maladaptive phenotype that we've seen before, suggesting, yes, that this is an effective way to deliver estradiol in the brain and provides an option that is more viable for human use. So our working model that we have so far is that in intact mice, we have the normal production of testosterone and conversion of testosterone to aromatase, conversion of testosterone to estradiol through aromatase. And then estradiol binds to the estrogen receptor beta in the basolateral amygdala, and then we have this increase in activity in the nucleus accumbens. And this is happening in response to rewarding stimuli. So in orchiectomized mice, so in mice that they don't have any testosterone production, then we don't have any conversion to estradiol. And what we've seen is that on this scenario, we've seen that we have a decrease in activity, which results in this stress-induced maladaptive phenotype related to depression. So what our conclusions are is that estradiol has a causal relationship and plays a seminal role in male stress-susceptibility in an animal model that is relevant to depression, and that decreased estradiol and not testosterone mediates susceptibility to stress in hypogonadal males through its action on the estrogen receptor beta. And then, importantly, we provide evidence for a novel approach by using trace-selective estradiol delivery, which we will avoid the undesirable peripheral estradiol effects. And it's a more viable treatment for human use. Lastly, I want to thank all the people that were involved in this project, Todd as my PI, and all the people in its lab that throughout the years helped, Brian and Michael, and also Peg from the pharmacology department, and Ishvan from the epidemiology and public health department, as well as our collaborator Laszlo and Vian for the design of the DHED, as well as the funding supporting this project. Thank you.
Polly, really impressive body of data, real tour de force. This is, of course, I've seen it coming along and seen presentations, but this is the first time I've seen it all together in a very formal presentation, and it's really impressive. So at this point, we can ask questions through the chat, or also feel free to unmute yourself and ask questions directly to Polly. And so, Polly, I have one question. Just going back to your early slides and the meta-analysis that you were discussing. Are those males that were included in the meta-analysis where they were treated with testosterone? Are those males that are hypogonadal, or are they just males with normal levels of testosterone that were given testosterone supplementation as a treatment for depression?
They were hypogonadal. They specifically targeted in most of the studies middle-aged and aging populations that they showed this decrease in testosterone production.
Thank you.
So, Polly, that was a great presentation. I was trying to understand what testosterone dose is given to the mice to actually reverse their phenotype. Is that a dose that would be comparable to what is given to humans? It's translated to that level, or so it's more in the high dose level or the low dose level?
It was more at the high dose level, which is something similar that we've seen in humans also, like the most effective testosterone effects are with the highest doses rather than the lower. But in this occasion, I went with what was published in the literature, so we haven't measured any plasma testosterone levels to make the associations, but based on the literature, we are on the higher end.
Okay. And the aromatase inhibitor that you're using in those experiments, was that letrozole or?
Yeah, it was letrozole, which we know that causes the blood-brain barrier because most of them actually they don't. But yeah.
Polly, there's a question in the chat from Leon Brown.
Yes.
And the question is there any potential role of ER alpha for other estrogen receptors other than the ER beta that you pursued most vigorously?
Yes, so for the ER alpha, we did find some effects with the social avoidance, and also there is a lot of literature investigating the role of ER alpha in female depression. And yes, definitely ER alpha does play a role, at least in female depression. Now, the G-coupled protein receptor is a newer receptor, so we don't know a lot about it yet, but some preliminary reports in animals may suggest that also might have a potential role, but for now, it's not very clear on the G-coupled receptor. But ER alpha, in terms of the female depression, for sure, yes, it has a role.
Just one more question, Polly. The letrozole story in the depression, the depressive phenotype, do you see the depressive phenotype in other tests or in just by letrozole, or is it primarily with the social defeat?
So in our controls, we know that at least at baseline, without any stress, we don't see any social avoidance or any anhedonia phenotype, and also sucrose preference, which again is anhedonia measure, and we didn't see any differences. So just treatment with letrozole, we don't see anything. So it seems that like we need a two-hit hypothesis to reveal the phenotype. So we need a second factor that will come in and reveal these effects.
Okay. Thank you.
Any other questions? No need to be shy.
Okay.
Thanks again, Polly, for joining us in Grand Rounds and giving this presentation. I hope in a period of time the potential to see a new medication for treatment of depression in hypogonadal males, whether it's the D-head molecule that you worked with or potential molecules may improve upon the some of the properties of D-head. All right, I think that concludes our Grand Rounds for today. Thanks again, Polly, and thanks for everyone for joining us.
That was really great, Polly. Thank you.
Thank you.
Thank you, Polly.